Hello, I'm Paulo Garcia, and I am CEO and co-founder of Kytopen. Imagine a world in which what you do at discovery stage scales seamlessly through development and into clinical manufacturing. That is exactly the type of future that we want to enable for you, our therapeutic partners that are doing great work in advancing cell and gene therapy. Today, I'll introduce you to Flowfact, our non-viral gene delivery technology that does just this: facilitates discovery, enables optimization through process development, and into clinical manufacturing without limitations in terms of scalability. We all know that in these advanced therapies, the cell is the therapy, but the genetic material needs to be introduced into the cell in order for us to be able to enhance their functionality and achieve all of those therapeutic responses that we are striving for. There are many technologies out there that use viruses and non-viral solutions in order to introduce the genetic material into the cells, and these involve electrical energy, mechanical energy, even nanotechnology. All of these technologies have advantages and areas of improvement. Some of the things that we need to make sure the delivery technology achieves is cell functionality, therapeutic cell yield, and be able to scale from the clinic through commercialization. The Kytopen technology is able to achieve all of these three tasks. Our mission is to collaborate with you, therapeutic partners, in order to improve the lives of patients by using our automated cell engineering platform. Flowfact is a non-viral delivery technology that has the ability to scale out, and that is high throughput capabilities, and also scale up, and that's for cost-effective cell therapy manufacturing. I'm extremely happy to share with you our beta device for the large-scale manufacturing that has been built with flexibility in mind. Here you can see that the sample is driven by a peristaltic pump in a completely closed manner from an input vessel with the cells and payload of interest into an outlet vessel, which is the bag containing the growth media. The magic happens in the center cartridge that contains our flow cell, and this is the area in which cells are exposed to electromechanical energy in order to gently disrupt the membrane, introduce the genetic material, and allow those cells to go through into recovery very quickly. We have adapted a liquid handling system in order to enable high throughput optimization of manufacturing conditions. And here you can see the sample being aspirated. When it is dispensed into growth media, that's when the magic happens. That's when the cells experience that electric field that is mild and the mechanical energy from the fluid flow. We leverage this capability in order to complete optimization screens in less than 10 minutes for an entire 96 well plate. This allows us to identify the regions in which the yield of engineered cells is maximized, and then we can transfer those same parameters into the large volume manufacturing platform for engineering your cells at scale. The flow cell is really the secret sauce of our technology, and it is the same electrodes, the same material, the same geometry, the same flow rates, and same electric field in both platforms. That's why our technology is scalable, because we are not building something larger. It's continuous fluid flow that is laminar in nature and is very gentle for the cells. Same flow cell in both platforms. We have generated some compelling data in very relevant cell types, including stem cells, CD34s, and iPSCs that retain their stemness, their functionality, and the ability to be differentiated into different lineages. Additionally, we have processed T cells with our activated or non-activated, and I will share some data in further slides. We have also demonstrated the power of our technology in B cells, in natural killer cells, both unstimulated and activated, and in very delicate cell types such as monocytes that are fragile to manipulate with other technologies. In every case, when we try our technology, we are able to achieve very high transfection efficiencies with very low impact in cell viability compared to the non-treated controls. An example of this type of data is shown on the screen, in which our scientists manipulated primary T cells and co-delivered two types of mRNA payloads. In this case, the payloads are coding for an mCherry and a GFP protein. You can see on the top right quadrant of this flow cytometry plot that there is a one-to-one correlation between the expression that we observe, both at around 95% efficiency with minimal impact in cell viability. This starts generating the potential for multiplex editing, and those are ongoing efforts at the company at this stage. Additionally, we have manipulated naive T cells. These are cells that are generally hard to transfect, and what I want to point out is that on the top right plot of the figures, the viability of these cells is not impacted at all. We have greater than 95% cell viability compared to the non-treated controls. And on the bottom left, we have greater than 95% transfection efficiency. This means that we do not have to compromise between the viability and the efficiency of our delivery. We can maintain high levels in both metrics. To take it a step further, we have also demonstrated that the markers for naive T cells, CD45RO and CD45RA, are retained at 24 hours after the transfection occurs. These cells were 100% naive before going into the Flowfact technology, and they remain 100% naive at 24 hours when we did the analysis. Finally, one of the things that we are really excited about is that the cells can be manipulated immediately after being processed. What we're showing on the bottom right is the ability to activate the cells after being processed, and the cells exhibit identical growth curves than the non-treated controls. This means that these cells retain their functionality, they can divide and expand, which is needed for achieving the high cell yield that is needed in a therapeutic program. The way we are collaborating with partners is by offering an optimization of your particular cell payload combination. This is by leveraging the Flowfact array technology. We have capabilities of optimizing the parameters in an automated process that is less than 10 minutes to complete an entire 96 well plate. The data that results from this optimization is depicted in this heat map, in which each of those unique parameters are demonstrating the yield of engineered cell therapies. In this case, the input was 1 million cells, and you're seeing a region that is quite broad, in which the yields, the resulting yields, are order 90% from the input number of cells. What we do with partners then is to make sure we verify the scale up, and we do this with the TX platform. In this particular example, we're demonstrating the ability to scale up from 1 million cells to 250 million cells. The data is normalized on a per 1 million cell input, but what you can see is that there is equivalence in terms of the yield of engineered cells in terms of the input material. We demonstrated this in multiple donors, but at the end of the day, this is demonstrations that are done at Kytopen. The real demonstration happens in your hands with our beta device, and that's where you can use your in-house facilities, your process development scientists, and demonstrate to yourselves that the Flowfact technology is a gentle way of delivering genetic material into cells in a completely closed manner and with flexibility in mind. We provide visibility into this optimization, and we select the parameters together in order to define what steps to take into the clinical manufacturing. very excited to share with you one of our latest data sets that we achieved in collaboration with a therapeutic partner, specifically around primary natural killer cells. In this case, we leveraged the array platform to do an optimization of CRISPR-Cas RNP delivery into the cells. We optimized the cell processing conditions, the RNP amount that was required to generate expression, and the Flowfact parameters. Again, the heat map that you see are 96 unique combinations of waveforms and flow rates that we programmed into our technology in order to maximize cell yields. In collaboration with the partner, we look at the viability, and these viabilities are greater than 90%, and we look at the knockout efficiency by surface staining that are, again, greater than 90%. The black boxes that you see in the heat map are the 5 parameters that we decided to do further verification with the partner in order to generate replicates and also subsequent scale-up. We are extremely proud that that technology is able to maintain high cell viabilities while achieving high knockout efficiencies. We do not have to compromise between those two very important metrics. Here, what we're showing is the confirmation studies that were performed across 3 different donors with the 5 different Flowfact parameters that we selected. Across all of these donors, we were able to maintain the high cell viability and the high knockout efficiency that we identified early on, with equivalent performance across the multiple donors for the first 2 waveforms that we tested. This is really powerful because we are able to now provide the partner with an optimized protocol that they can take for their manufacturing conditions using the Flowfact TX manufacturing platform. We have assembled a world-class team in terms of the management in order to build and continue developing the therapeutic relationships with our partners with expertise in operations, product development, commercialization, and microfluidics. The TX development roadmap this year is all focused on our Flowfact TX beta device. In this case, we are doing beta testing with early access partners that will go through the first semester of this year. And we have already kicked off our efforts in order to generate a GMP solution by the end of 2021. The GMP solution will encompass a GMP system, GMP consumables, and GMP buffer so that we have the full suite of solution for your clinical needs in 2022. The goal is to be able to demonstrate clinical efficacy with you in 2022, and then take all of those learnings and incorporate them into the next generation Flowfact TX version 3.0 to continue helping patients that are needing these life-saving therapies. As I mentioned, last year we unveiled our beta testing for the Flowfact TX platform. We generated some seed funding in order to advance these efforts, and this year we're laser focused on achieving cell therapy manufacturing partnerships with therapeutic companies such as many of you. This will be done in different cell types, including T cells, natural killer cells, and stem cells. And the goal is to be able to provide a solution by the end of the year that can be implemented into your clinical programs next year in 2022. There's several conversations that we have ongoing, and the exciting aspect is that the need is there and the technology is quite compelling in order to accelerate your timelines into the clinic and be able to reduce the cost of manufacturing of your important cell therapies. In summary, we want to share with you that we have a technology that generates delivery with seamless scalability, and we can optimize the manufacturing conditions with low volumes of tens of microliters, and enable cell therapy manufacturing with large volumes that are greater than 10 milliliters. The flow cell depicted in the center is our main character, and here we can demonstrate scalabilities of 100 to 1,000 times scales in order to demonstrate clinical manufacturing that is compelling for your therapeutic program. In just 1 minute, we can process greater than 1 billion cells. And the fact that our technology is continuous flow means that if you wanted to process 10 billion cells, all we have to do is introduce a larger volume upstream and flow for a little bit longer. The technology scales naturally, and that is why this is going to help you in your programs in order to help many more patients that are needing these cell therapies. With this, I would like to close and thank the entire Kytopen team for the dedication in terms of developing this platform, demonstrating our capabilities with cell types that are hard to transfect, and also our investors that have believed in us from the beginning and that continue to provide support and guidance along the way. Lastly, I want to thank our manufacturers in order to help realize the vision of a GMP solution that you, our partners, can leverage in your own programs. Thank you for your time, and looking forward to many discussions in this conference in order to generate some collaborations for the future and help many more patients. Thank you again.